But I put it back up here because I want to talk. That's the bacterial genetics basic discipline. It was all the advances in bacterial genetics and the studies done with bacteria and viruses that really led to the field of molecular biology. And eventually, using those microorganisms and those genetic techniques with microbes, it also led into the development of the field of biotechnology. So the importance of the basic studies in bacterial genetics really laid the foundation for these two additional disciplines. In fact, our department in which microbiology is taught is called the Molecular Biology and Microbiology Department. We renamed our degree BS in Biomedical Sciences, even though it has a strong molecular thrust to it. You know, we teach Molec 1, Molec 2, molecular immunology, and so on. We renamed it just to better reflect the broad area because we teach human physiology, human anatomy, pathology, a wide variety of courses in the biomedical sciences, in addition to microbiology, immunology, and all the other areas. So these two fields really, and molecular biology basically is the foundation for the techniques used in biotechnology. So basically from bacterial genetics, we developed these two new fields that basically helped us move the focus of microbiology more to molecular microbiology, which has led to biotechnology, genomics, proteomics. You know, we're sequencing, we've already sequenced the human genome, we've done worms, Drosophila, and everything else. We even did one of the guys that won the Nobel Prize for the double-stranded helical structure of DNA, Wilkins, not Watson and Crick, but Wilkins. So this whole, in the 1900s, all those studies with bacteria, simple things like those of you who had genetics know about the pneumococcus experiment showing that DNA was the genetic material. The Hershey-Chase experiment in which they showed that when a bacteriophage infects a bacterial cell, it inserts its DNA and not its protein, so it has to be the DNA that's genetic material. So all of those studies led to these two fields. Okay, so what is the focus of microbiology today? Well, in the book and in any course we teach in microbiology, there's going to be two broad focuses, and I don't want you to miss those. One we've already talked about, that we use microbes as model systems to study basic life processes. So all cells have much in common. Microbes are just a nice, simple type of cell to study. And if you recall, I said, yes, eukaryotic cells and prokaryotic cells are different. Our bacteria are prokaryotes. Our archaea are prokaryotes. And there are distinct differences between prokaryotes and eukaryotes. But if you look at the basic cellular processes, they're very, very similar. I can tell you that transcription involves taking one strand of DNA and making a copy of that nucleotide sequence in messenger RNA. That's called transcription. Transcription is common in eukaryotes and prokaryotes. Do the details differ? Yes. You're going to learn when you take a course in molecular biology or genetics that there's a capping that occurs with eukaryotes. There's a poly-A tail that is, that's put on in eukaryotes that aren't done in prokaryotes, but still the basic mechanisms are the same. The way DNA replicates is very similar. Just some of the minute differences will be something you'll learn in a more advanced course in molecular biology. So it helps us study all life processes, but more importantly, the second theme we have is apply that knowledge, the knowledge about microorganisms that we'll learn in this course. Everything's not moving, is it? Here we go, okay. For the betterment of humankind. So we're going to solve practical problems in medicine. We've already talked about this a little bit, the development of antibiotics. We're going to talk today a little bit, if I get to it, how Koch was able to develop some methodology or postulates to show that a microorganism is the cause of a specific disease. We talked about agriculture, making sure you plant put in the ground plants that can enrich, convert that nitrogen to ammonia that plants can use, how you alternate that so that you run the leguminous plants one maybe season, and the next season you run something else because it's already been enriched in ammonia, but then the part that's used for other plants, you now plant the legumes there. Industry, all the biotechnology techniques that can make all kinds of products for us, and we'll even talk about this in the next slide because we're going to talk about something that happened, it's probably a little more than 30 years, about 1980. So we're looking at about 33, 34 years, pushing 34 years now. When microbiology hit Wall Street, 1980, that was over 30 years ago. Thousands of buyers rushed to purchase the stock, and every time, I guess when Google went on, and thousands of people went purchase stock in Google, but that was the excitement back then. They were all rushing to purchase a stock in this new obscure company. It decided to go public. Price per share increased 150% in a matter of minutes. Okay? And if you look at it, it certainly wasn't an oil company. It had only 140 employees, no track record, very speculative. Why would people be rushing to purchase this company? And that company today is still called Genentech Incorporated. It was one of the first biotechnology companies. It produces products like human insulin, hepatitis B vaccine, interferons, and so on. Genentech still exists today. Its name came from the term that describes that science, genetic engineering technology. So the gen from genetic, the EN from engineering, and the tech from technology. And that's how they came up with Genentech. If you look at genetic engineering technology, basically it's applying gene technology or recombinant DNA technology to develop lots of products that are useful for mankind. That means we're going to take genes from that we want to have that product from that gene. For example, there's a gene for insulin, or one of the changes in the gene for insulin. You're going to want to clone that gene and actually have it make human insulin. You actually need to do two genes because you have to have the A chain and the B chain. But simplistically, you say you clone the gene and you have a microbe make large amounts of it. So a little more technical, you transfer defined genes into rapidly growing microorganisms, and it's possible then for them in an in vivo system, meaning inside that living organism, they can make thousands of products useful to mankind. Because whatever gene we put in, it's transcribed and translated into the polypeptide chain. So if you clone an insulin gene, that microbe can make it. Now, the thing that's key here, it says rapidly growing microorganisms, and that's something you may not still have a full appreciation for because we're early in the class. When we get to the section on microbial growth and reproduction, you're going to find that we're going to talk about how quick those microbes can reproduce. That's one of their most dramatic and significant properties. That's why if you have an infection that gets in the blood, septicemia, it's sometimes only a matter of 30 minutes before they go from like maybe 500,000 to maybe 10 million, because they can double every 20 minutes. And if it's one microbe and it doubles to two, that's only an additional one. Okay, when you get up to 500,000 and it doubles, you add 500,000, you're at 1 million. And then the next 20 minutes, you get 1 million more. So that's why people sometimes can die within 30 minutes if they don't get to the hospital if they have septicemia. Because those microbes, once they're in a high enough number in the bloodstream, they go crazy. They reproduce so rapidly. So if they can grow and reproduce that rapidly, see, they're going to transcribe and translate their genetic information. They're going to go from a few cells to billions of cells in a short matter of time. And they can pump out tons of that protein product that's cloned into the genes of that microorganism. And so it's very important to understand just how rapidly they can grow, and we'll get to that later in the class. We made all kinds of things that are useful to you. We made vaccines like the hepatitis B vaccine. We've cloned human insulin, so we make human insulin by these biotechnology techniques. Antiviral agents like interferon, okay? Hormones like human growth hormones, somatostatin, all kinds of agents that they can be useful in either prevention of disease or treatment of disease. So if you really look at the whole, this new field, biotechnology uses genetically modified microorganisms. That's not the genes they normally have. We put new genes in, and they synthesize product typically of high commercial value. Sometimes it's not as much high commercial value, although that's what the companies want, because they want to make money off these biotech companies do want to make money, okay, because they have to pay for everything and they have to pay salaries. But a lot of times they do make some products that really they don't recoup all their funds because of concern for human health and so on. So biotechnology uses the tools of genetic engineering, and the big advantage, once again, it makes a lot of the gene product. Because if that microbe can multiply so rapidly, it can transcribe and translate those genes at such a rate it can produce tremendous amounts of product. You can grow them in a big fermentation vat, tons of microbes, put out tons of the product in a very pure form. And this is pointed out by two examples. If you look at interferon, We used to get interferon. It can be used in antiviral therapy, can use in cancer therapy. It used to take us 100 liters of human blood to make 1 milligram of interferon. Now, it's not that you get 1 milligram of interferon freely from 100 milliliters of blood. You've got to go into that blood, which has all kinds of other proteins, and what's one of the common proteins in blood? Hemoglobin, high concentration. You have to purify the minute amount of interferon from all those other proteins. It's expensive, it's costly. Interferon is just a tiny amount of protein in blood. Today now, we can make that same amount, 1 million of interferon, just from a little liter of E. coli. Just growing a little liter of E. coli in a flask, we can get as much interferon if we've got that gene cloned in the escherichia. Another good example is human growth hormone, somatostatin. They used to take 100,000 sheep brains to make the same amount of somatostatin that we can now make with just 2 liters of E. coli grown using the genetic biotechnology techniques. So you see, it's been a tremendous advantage, the development of the whole field of biotechnology, using microbes and basically as little factories to make the products that are useful to mankind. Okay, this just summarizes a lot of the things we talked about before. If you look here, we talked about it. I think, and I have to apologize, guys. I hate teaching 2 sections of the same course because I like to give examples. Not every example's on the PowerPoint. So sometimes I'll forget to give you an example and I'll give it to the other class, and then when I come here I'll give you an example I don't give to them. So if I repeat or if I forget and say, well, I told you this before, if I didn't, I'll just ignore me because it's kind of hard to keep them all straight. But anyway, we already talked, I think, about Swiss cheese, how it's due to the propionic acid fermentation. The taste is the propionic acid, and the little gas, CO2 evolved, makes the holes in Swiss cheese. So we use microbes to make food products, our alcoholic beverages, sweet acidophilus milk, all kinds of products. Sauerkraut from fermentation of cabbage. We talked about biofuels and corn being converted to ethanol. I didn't talk about bioremediation, I don't think. For example, we can, the oil spills, we can put on those oil spills microbes that degrade hydrocarbons, and that's one of the things they do. So I'll show you here, bioremediation. Use of living organisms to degrade pollutants in the environment, okay? And good example, bacteria used to degrade oil and oil spills. They have these hydrocarbon oxidizing enzymes that break down and oxidize that fuel. We also use bacteria to degrade polychlorinated biphenyls and that trichloroethylene. These are used in dry cleaning. So we have problems, those contaminate the environment. So bioremediation is another thing I haven't talked about before that we use microbes for. Talked about the cow and the rumen of the cow and how it wouldn't be able to degrade its cellulose. So the microbes in the gut or the rumen of the cow do that job. I think they talked about that in the film also that you saw the other day. Nutrient cycles, we talked about the nitrogen cycle. There's also a sulfur, carbon cycle, and so on. So microbes play big roles in all of these. And even treatment and cure and prevention of diseases. They help us, microbiologists identify new diseases. There was a microbiologist that identified the Legionnaire's disease bacterium. Took him a long while, but he found it when he was cultivating some of that tissue, trying to look for rickettsia. It turned out it wasn't a rickettsial microbe, it was a bacterium. It was a very fastidious bacterium. He had to grow it chocolate agar, which he used for rickettsia, and in all those tissue samples, he kept seeing this big rod-shaped bacterium. It turned out that was the one that was the causative agent in the Legionnaire's disease. So he found it by accident, looking for something else. So anyway, this is just to show you applications, just as a little pictorial representation. Oops, here we go. I go too fast here. Okay, bioremediation we covered. Okay, we're going to switch a little bit. We're going to look at properties of living cells. Okay. We're going to be looking at microorganisms in this course that include bacteria, archaea, and even a few eukaryotic microorganisms. And all of these are cellular. In this course, we also talk about some acellular entities. That means they do not, for example, viruses, viroids, and prions, they don't have cellular structure. But we're talking about living organisms. We're talking about those that basically the cell is the basic unit of their structure. All cells have a cell membrane or a cytoplasmic membrane that basically defines the compartment of that cell. Okay? But if a cell doesn't have a cell membrane, it's not a cell. You lyse the cell membrane, that cell no longer exists. You put red blood cells in water, what's going to happen? They have no solid concentration. Okay, water is going to, basically is going to move in. I mean, even the water has no salt concentration. The blood cells do. The water moves in, lyses the red blood cells. They no longer exist. Right? So you need to have a cell membrane. So we talk about invariant structures as structures or components or macromolecules. Even we can just say invariant structures or molecules. All cells have it. All cells have a cell membrane. All cells have ribosomes. Now, the ribosomes of eukaryotes and prokaryotes might be different, but once again, they do have ribosomes. And both ribosomes consist of a little small unit and a larger unit, a component. So if we look at bacteria, we see they have 70S ribosomes we'll talk about later. Eukaryotes have 80S. Now, I'll tell you what that means later, but they're slightly different, but still all living cells have ribosomes. They all have DNA as the genetic material if we're talking about cells. And if we're talking about viruses that are not cells, that are acellular, we can have RNA as the genetic material, either single-stranded RNA or double-stranded RNA. But all of these cells can have both DNA and RNA. Variant structures are structures that not all cells have. For example, mitochondria, the great majority of prokaryotes, almost all prokaryotes, I think there's one exception, don't have a mitochondria. So we typically say prokaryotes do not have mitochondria. Eukaryotic cells have mitochondria. Okay? Chloroplast. Photosynthetic eukaryotes have chloroplasts. Non-photosynthetic eukaryotes do not have chloroplasts. Bacteria that are photosynthetic don't have chloroplasts. They have the same functions. They can carry out light reactions, but they don't have that entity, that membrane-bound entity called the chloroplast. So these are what we would call variant structures, not found in all of these cells. We look at the characteristics of microbial cellular life and look at the characteristics or properties pretty much found in all cells. In contrast, some characteristics found only in some cells. These three here are very important. Metabolism are all the chemical reactions of the cell. We'll talk a little more about that. Reproduction, also regeneration, has to do with reproduction of the cell, repair of the cell, replacement. And evolution basically is a characteristic of living cells. They can change over time. They can mutate. The cell can mutate. Whether you believe in the origin of life as having evolved, people having evolved and so on, I can tell you that evolution does exist with a lingual, because I can take a microbial cell that is penicillin sensitive, that is destroyed by penicillin, can't grow in the presence of penicillin. If I have a big population, I'm growing them, and I dump some penicillin in, there will be a mutation. Even though the rate is rare, there will be a mutation that will mutate to penicillin resistance. And what's going to happen if it mutates and it's in penicillin, what's going to happen is it's going to continue growing and all the rest aren't. So I take a sample out and put it into another flask that has penicillin. Okay, those that are penicillin resistant that have reproduced are going to all be favored. Pretty soon I'm going to end up with a population that is completely penicillin resistant. And that's what basically evolution is. We have a cell, and now it has changed. It is now, it was penicillin sensitive, it is now penicillin resistant. So even die-hard people who do not believe in evolution of people from earlier, simpler organisms, got to say that at least it exists as cells. Okay, we can do that in the laboratory. Okay? So anyway, evolution characteristics of cells do change over time. We know that they evolve. Natural selection, acting on variations caused by mutations, makes them change. So if you get the mutation and there's no penicillin there's no selective advantage. So you're not going to get that particular property, that organism to predominate. Okay? You have to have the selective pressure. You have to keep the penicillin there so it favors that microorganism that is mutated. And that's why you get it to evolve. So what happens is mutation is the raw material. And natural selection, some selective pressure favors that mutation and allows it to propagate. Okay, we'll talk about the others in just a minute. Here's the little diagram from the book. It pretty much explains everything here. If we look at properties of all cells, compartmentalization and metabolism, meaning you have a spatial cell membrane that basically makes the spatial part of that cell, and you have chemical reactions occurring. Metabolism basically is going to be all the chemical reactions in the cell. we're gonna be talking about these when we get into chapter 5 on microbial metabolism.